Chakraborty on dais. A very good morning to you, sir. Now I request our respected Principal Maharaj, Swami Kamalasthanandaji, to felicitate our guest and deliver the welcome address.
Respected Dr. Debojyoti Chakraborty on the dais, in front of me his beloved father and mother, and my dear colleagues and very dear students present here. So we are very happy to organize this seminar today. The speaker, after that my colleague will introduce you. And I just tell few words regarding this seminar. He is a good student and good researcher in field of genetics and molecular biology. After that, when you heard that he is a great biodata, he is the one of the 30th scientist in European Molecular Biology Organization. And last year, I think everybody, those who go through the news, you can understand this Feluda kits for COVID detection. First indigenous COVID detection kit Feluda, which is actually come from IGIB, Integrated Genomics, and this Integrated Biology, Institute of Genomics and Integrated Biology, this lab. And he actually the principal scientist in this project. So I do not tell this, I only tell this, he is a great scientist in one side. And another side, he is a accomplished and professional musician. Accomplished and professional musician, not amateur. He has a lot of prizes, especially he has performed the Doberland Music Conference. Those are actually little bit know about Doberland Music Conference. It is a elite platforms where not amateur party they could not enter. This is very prestigious platform where the accomplished musicians and performers they can perform there. And he performed there. And Doberland Music Conference every year organized till now, and those are actually interested about this information, they can understand what type of musician they participated. So he also very kind enough, after this today's session, after 2 o'clock he actually performed his Setar recital. Maybe you are not present, others will come, but we organize this program, his two performances, one is his great scientific achievement, another his musical acumen. So with these few words I also tell all you that his talk will be something different, very interesting. Before talk I can guess, you freely interact with him, but time is very short, because with 12:30 we have to finish, because another session we have to again start. So with these few words, once again I welcome him heartily from this organization, from all of you, from all faculty members, for coming over here after taking all type of odds and difficulties. Once again, thank you, Dr. Debojyoti Chakraborty. So I once again thanks all of you. Thank you.
Now it's time to formally introduce our today's speaker. I feel really excited to coordinate today's seminar and introduce this exceptionally talented Dr. Debojyoti Chakraborty. I would like to request all the students to carefully listen to it because it's not only fascinating but also inspiring. Dr. Chakraborty is officially a PhD from molecular biology from the Max Planck Institute, Germany, and presently working as a principal scientist at Institute of Genomics and Integrative Biology, CSIR-IGIB, New Delhi. His pioneering work on genome editing has led to the discovery of COVID-19 detection kit Feluda. You probably all of you know because it was so highly circulated in all the print and the news channel that time. If I remember correctly, it is FN-Cas9 edited limited uniform detection assay, the full form, and is coming from, it's a CRISPR dependent system, and this Cas9 is derived from the Francisella novicida, if I am not wrong. This technology brought to the society by the Tata, and this technology is awarded CSIR Technology Award in 2021. Dr. Chakraborty is also the recipient of Young Scientist Award from INSA, Indian National Science Academy, Mark Tata, Lady Tata, CSIR, Vasvik Trust, etc. He has become one of the 30 scientists across the world to became the member of the European Molecular Biology Organization, EMBO. His current research interest is on prospects of genome engineering to cure the hematological disorders in tribal population in India. It's not end here. It's time to introduce him in a completely separate aspect, the musician Debojyoti Chakraborty. He has been learning sitar under late Sri Sunil Saha and Pandit Devaprasad Chakraborty for the last 28 years. He has performed in India and Europe in several concerts and awarded Best Instrumentalist Award in Dovernan Music Conference Competition and West Bengal State Music Academy, among many others. He is a recipient of National Scholarship in Music from Ministry of Human Resources, Government of India. He has also contributed to the academic research related to the mathematical modeling of Indian classical music and featured in almost all the print and broadcast media in India and abroad too. So I am very much privileged to welcome Dr. Chakraborty to give his today's lecture. Thank you, sir.
So good afternoon, everyone. Thank you, Maharaj. Thank you, everyone, for this very nice introduction. I don't think so I'm really deserving of such a long introduction. There's a lot to do, lot to go ahead. So these are little bit of, so it's kind of undeserving, So today I'm going to talk to you about something which you might have heard, some of you might have studied. How many of you know about genome editing? Just raise your hands. CRISPR, genome editing, do you know? 1, 2, 3. Okay, few people know. So this comes from the fact that we know about DNA, right? All of you are from student of biology at some point. So you know that there is inside our cells, there is DNA, there is RNA, protein, and whatever is there in the DNA, যেরকম আমরা বলি কি DNA তে music আছে, DNA তে science আছে, এরকম বলা হয় না? So whatever is there in the DNA is kind of fixed in terms of how we express those characters, like your hair color is black or brown, or eye color is black or brown, and so on. But that also means that if there are some things in the DNA which causes diseases, let's say for example somebody might be prone to having cancer at the age of 70, or let's say age of 50, because it's there, runs in the family, okay, that is also there in the DNA. And almost it was thought even 20 years back that you could never change that. If you are born that way, you remain that way. Okay, if you have that mutation, if you have a problem in the DNA, then it stays like that. But what I'll tell you today about is a technology which allows you to change even that. That almost sounds like science fiction, Professor Shonku. So it almost sounds very weird that how could you change the DNA of a person and really change how that person becomes later on. So that is the technology which is genome editing, because it's genome and it's editing. You are correcting the mutation, and I'll talk to you about that today. So the title of my talk is Reading and Writing the Genome with Precision. Means how can you read the genome? You know, you can see the letters of the DNA. What are the letters of the DNA? Correct. ATGC and different combinations of it. And how many combinations? What is the length of the genome? No. 3.2 billion letters. 3.2 billion letters inside every cell. In that tiny 5-micron cell packaged inside a tiny nucleus, we have our DNA. So here we are talking about changing those letters. So reading those letters and then correcting them. That's what CRISPR does. So, you know, this is a picture not from Park Street but from a different metro station in Delhi. It's called Rajiv Chowk. Okay, and you see there is a lot of people who are waiting at the metro station trying to get into one single compartment. You don't have to go to metro station. If you go to Kolani line and you wait there at the local train, it's almost same, correct? Many of you might see that. And sometimes you do not have to make an effort to go into that bogie. You just go in and you come out. Right? It's a natural process. You just get pushed into it, you get pushed out of it. Okay. When I went to Japan for a similar kind of in a university to give a talk, I saw that in their metros there is also a lot of crowd. But then there are people who are standing there. They are called ushers. They actually push people in, and then there are people who pull them with, you know, very politely standing there, line in a queue. In some other place in Finland, there is no queue. There are more trains than the number of people. There's weird things all over the world. So all that I want to tell you is that there are different ways of dealing with the same problem. And sometimes even for science, like today if you want to look at the brightest and the farthest galaxy out there, can you do it with a normal telescope? You cannot. You have to build a telescope that is that powerful. So your questions would have to be, your questions have to be answered with the right type of tools. You just cannot take tool A and answer question B. Sometimes you have to develop the tools. You have to develop microscope. You have to develop biochemistry assays. Okay. So in India, we have different ways to solve that. Like, see this individual here. So you have heard about Murphy's paradox. And this is maybe a train traveling paradox. At same point, you are inside the train as well as outside the train. You can travel that way also, but it works. Okay. So in India, we have ways of solving that problem. In a different country, we have a different way of solving the problem. But the essence is, I will just quickly. Take it. So the essence is that we are all here talking about how to solve some of the problems in biology, and particularly in some cases in diseases where some of the technologies that are already present might not be enough to solve let's say, diseases. This particular disease that I'll tell you today about, it's called it's sickle cell anemia. Okay, just give me 1 second. I think it's the wrong presentation. Okay, yeah. So the at this point of time, like I told you that this is not it will come eventually, don't worry. It's it's not a science fiction to say that we have cured genetic diseases, because this individual that you see over here, the African American lady, her name is Victoria Gray, and she is the first person in the world whose DNA has been changed, okay, using CRISPR. And this doctor who is there, Haider Frangul, is the first doctor who actually administered it. Fortunately, we are also working with him. So this is a disease which has been cured using a genome editing technology, where the DNA of an individual has been changed, and it has also been done outside the body. You take cells out of the body, you correct it and give it back, or even directly inside the bloodstream. You can deliver it and you can do that correction. All these sounds a bit science fiction like, I know it's not so easily said as done, but I'll take you through the things step by step and how it actually works. So you have heard about CRISPR. You know that in the year 2020 there was a Nobel Prize that went to CRISPR, right? Who are the ones who won the Nobel Prize? Yes.
One is Emmanuel Charpentier and another is
Jennifer Doudna. Yeah, fantastic scientist, right? Women scientists. In the year 2020 was the year of women in science, in politics. Kamala Harris became the main advisor in the US. So fantastic things that are happening in women role in science and all. Unfortunately, I don't see too many women scientists here, but still. Just to tell you that CRISPR is something that really brought them to how well they have done, and the 2020 Nobel Prize was given to Jennifer Doudna and Emmanuel Charpentier for this technology. What does it do? It has this protein here which actually originated in bacteria, and this protein that you see in this kind of colored thing, it takes the help of an RNA molecule, and this RNA molecule is shown in blue, and this complex goes to any part of the DNA that you program it to go. Now, how do I imagine this? You imagine that you're writing a letter, typing a letter. Okay, you type this letter in the word processor file, Microsoft Word, and somewhere in that you have made a mistake in a spelling. Instead of writing the, you have written AHE. Okay, so how would you correct that? You take, sorry? Right, so you find and replace. You take your cursor there, right? Then you press a delete button, remove the A, put in a T, and then what do? So that's done. You save the file, right? So this works exactly the same way. So this particular protein complex is like that cursor. If you tell that RNA that I have to go to my BRCA1 mutation or P53 mutation in the DNA, by putting that sequence of that DNA on that RNA piece, this RNA goes to the DNA and forms a base pairing, hybridization, hydrogen bonding, okay? And then this Cas becomes active, a small piece of DNA to get incorporated at that cut. Then you could essentially change the letter that was originally there. Now how beautiful is that? Because if there was a disease letter, you could change that into a normal letter, correct? And that is the essence of genome editing. However, this is just to show you that all these different things actually happen at a time scale of a few seconds. So what you will see in this video is a Cas protein sitting on this strand of DNA. This is a DNA, this is a protein, believe me, okay? And you will see it takes only about 30 seconds for this Cas protein to, there you see, the cut, okay? So imagine you are putting all this Cas protein inside the cell, trying to cut and then repair. If in the process it goes to some other part of the cell, that's not very good, right? So you need to have a very precise Cas. You have to have a very precise machinery. It's like you take a paracetamol and it, instead of going and reducing a fever, it goes into the pancreas and does something else. So those are called drug off targets. These are called genomic off targets, genetic off targets. So that's not very good. Somebody told already there are 3.2 billion letters, and here we are talking about finding one letter in the 3.2 billion letters. Crazy, right? You have to have it very, very specific. So many people around the world, including us, we are working on trying to make this system as specific as possible, as perfect, as accurate as possible. So the postman, if it tells that you have to go and deliver this to, Ramakrishna Mission Rahora, it doesn't go to Ramakrishna Mission Narendrapur. Okay, that address this postman has to know very, very accurately. And how do we do that? We do that by protein engineering. We try to change the protein, the original protein which comes from bacteria. We try to change it. We may try to make it better and better and better so that it becomes so specific. So to cut a long story short, today I'm going to tell you that, in 2019, we first discovered, this is of course my colleague, my friend as well, he's Dr. Shobhik Maiti. Most of the work that I will tell you today about is actually also done with him. He's a biophysicist. So he comes from the field of understanding how protein DNA interacts and so on. So we figured out that sometimes you can look in nature and find out more efficient varieties of CRISPR proteins. So 50% of bacteria in the world have some form of CRISPR system or the other. And we looked at a bacteria called Francisella novicida and found that this particular Cas protein is very, very accurate. How do we know it's accurate? Has anybody of you done agarose gel electrophoresis? How many? Very good. You know, this is an agarose gel electrophoresis picture, simple picture, okay? What you see over there is that if you take a DNA piece and you take the Cas protein, then the Cas protein cuts the DNA. So you see those two bands 9, which is Streptococcus pyogenes Cas9, it actually cuts these off targets as well. But the ones that we kind of characterize is called Francisella novicida Cas9. It doesn't cut these off targets, it only cuts the on target. So that was very remarkable. We thought that, well, we knew that we have now a Cas which was so specific, so accurate. Can we use to cure diseases? And we had a disease in mind, of course, because sickle cell anemia, thank you. So sickle cell anemia is a disease which is very prevalent in India, okay? Sickle cell anemia, the cells, RBCs, red blood cells, which are normally circle, they become like a half moon. And children die because of it. Lot of them, several of them die. There is stroke, there is pain, there is crisis and all these things. And if you see, interestingly, most of the people who are affected by this disease are in the tribal population. These are the poorest of the poor people, okay? No access to healthcare and so on. So if we want to, and it's caused by a single mutation in the DNA. This GAG gets converted into GTG, okay? Due to which the blood becomes sickled. So if we could correct it back, GTG back to GAG, we would essentially get the disease cured, right? But for that, this Cas has to be active at that site. But unfortunately, we found that this Cas protein that we are working with was very good in specificity, but it was not very active. And it was way below here. If we had to increase it, if we had to use it for clinical therapies, we would need to increase its efficiency, efficiency of what it can do. So what did we need? We needed a higher efficiency variant, but most importantly, it should not change that high specificity it already had, because that is the USP we have. So you do protein engineering to make a protein more efficient, but keep the same accuracy. So we did a lot of different experiments. You don't have to go through all this. Basically, we put a single piece of DNA, stretched it between two beads and looked at how a single protein sits there and so on. And from there, we calculated that you could find specific places in the protein that you could mutate and still make this protein way more efficient. And we call them as engineered FN-Cas9 protein, EN-FN-Cas9. This was all done in Tokyo with one of the discoverers of the crystal structure of CRISPR in the first place. So now we had a protein which essentially does what we want it to do. It has high specificity and it is more efficient than the wild type variety. And it is not only efficient in the test tube, but it is also efficient inside the cells. So it can now, be used for very good editing rates and so on. And we solved the structure of this protein, figured out that this protein can actually bind to let's say, it can bind to more sites on the genome, due to which it can now target more diseases than we could normally target it. Okay, so its possibility of binding to different places increases. We went ahead from there and started making a toolbox of different types of FN-Cas9 variants, some to control the RNA, some to control transcription, some to control translation. I'll just give you an example of a very interesting project we run in the lab. So we actually use, something called mini brains. Do you know anything about mini brains, organoids? So imagine if you would take stem cells from an adult, okay, or from an embryo, and you grow them with specific type of factors in a 3D culture, then you could actually generate very tiny looking mini brains on a dish. So you would not have to take a brain out from an individual to test it, but you could actually make the brain on the dish. And this is what is being done over here. So you'd start with stem cells. Stem cells can grow into any cell of the body, and we tell them that you should grow only to the brain lineage. Okay, and this is how all these things are done. So we could engineer them, and specifically engineer them for mutations or diseases which you otherwise cannot study in humans. Example, autism, okay, schizophrenia, bipolar disorders. These are diseases which you cannot take out anything from the brain and study. It's not possible. Neither can you do it in a mouse, because a mouse is not a human. So cognitive problems, let's say cognitive problems means speech problems or being able to handle things like that. You know autism, how big this issue is, at this point of time. There are so many genes implicated. How to study that? So we've developed these kind of models which you can actually do on the Petri dish. You start with anybody's cells. You just give a drop of blood. From that blood, we convert it into stem cells. Later on, we'll discuss. Does anybody know about this? How this is done? It's called reprogramming. It got a Nobel Prize by Shinya Yamanaka in 2007, which says that you do not need to get the stem cells from which doesn't cause the neurons to form, as you see over here when you knock it out. So KO means knockout. So if you knock it out, these neurons do not form anymore from this brain. Okay, so the other thing that we learned was that, I told you about this high specificity of this Cas protein, which means that, you could not only edit the genome with very high efficiency, but you could also read the genome. That, if there is a wild type versus mismatch, you remember the agarose gel electrophoresis? That if there is a point mismatch also, it doesn't bind, it doesn't cut. So this not binding is shown here by these binding curves. KD means is an inverse of binding affinity. So higher the KD means that very little affinity for DNA to bind. So we see that when there is a mismatch, this particular Cas protein doesn't bind in such cases. So we thought, well, that's very interesting, because you could essentially use this property to diagnose disorders which are caused by changes in the DNA or mismatches in the DNA. And that is actually the origin of what you have heard as FELUDA. Okay, so FELUDA stands for FN-Cas9 edited linked uniform detection assay. I think some of you have heard about it in the media and all. And you all know why FELUDA is FELUDA, because we were two Bengalis in the team and we decided to call it through a Bengali name, because one of our competitors who was in MIT, Jennifer Doudna and Feng Zhang, they gave their names as Sherlock and Detector. Okay, and they were using different Cas proteins. So we thought, okay, we have to use our own Cas protein. Why not give an Indian detective's name? So that's why how FELUDA came about. And it essentially uses the power of CRISPR to diagnose DNA mutations. Okay, and this was it also got approval in India. Was the first paper strip test for COVID-19, CRISPR based, that was approved in the world actually for this. How does it work? It works on the principle that you have a Cas protein sitting on DNA, and if it binds strongly, then it can give some kind of a readout. But if it cannot bind, then it gives a different type of readout. And this readout is what we were actually exploring in the field. So here we used to go to different villages and look for how to diagnose sickle cell anemia. And if you see here, how typically this is done is via a very crude assay. They take blood, lot of blood, and then they do coagulation and then say that, well, the patient might be positive or negative, and so on. But you see that, for that you still need blood. And sometimes these little kids here who are standing there, someone like her, she's not very excited at the prospect of, having the finger pricked and then giving blood. So we came up with a strategy where we could simply ask them to spit into a small tube. And the same DNA that is there in your blood is also there in your saliva, right? So you don't need to have your finger pricked, and then there would be reagents that will break open the cells, and then there would be FELUDA reagents, and all this can be done within an hour or two without the need for sequencing. Which sequencing is how you read, and that is costly, that is expensive, takes time, long turnaround times, etc., etc. But all this can be done in the field. So that's what we were doing actually at that point of time with FELUDA. And then of course the government said, well, you have a technology, why don't you make it for COVID? And so that's why COVID came. And as you know, that FELUDA doesn't come alone, right? FELUDA has to come with? Correct. So you need Topshe and Jatayu, otherwise it's a sacrilege. So that's why we had Topshe, which is basically a smartphone app which can take a picture of that strip, okay? And it has been trained by machine language learning to tell whether there is a positive signal or not. And Jatayu, so Topshe stands for True Outcome Predicted via Strip Evaluation, okay? And Jatayu is a web server where you put in your disease of interest and it basically designs your FELUDA primers. And the full form of that is Junction for the Analysis of your FELUDA Experiment. Okay. And then, of course, all this went into, it had to be go into a commercial kind of a route. And this was actually given to the house of Tata. And Tata made a new company. They called it Tata Medical and Diagnostics. And they changed this all into a kind of a mobile device where they could put this whole thing in a robotic platform inside trucks and send it to different parts of India, because then you could multiplex this whole thing. So there would be robots which would be pipetting this Feluda reagents, there will be robots which would be reading this out, and you could do around 3,000, 4,000 tests at different parts where it could all go to. Okay, but we kind of deviated. We were talking about curing diseases. And as it turns out, when you have a technology like this and you want to take it to patients, it's a very long process. Typically, if you had a molecule and you would have to treat somebody with that, it is really a 30-40-year-old process, kind of a process, because you have to get regulatory clearances. You need a lot of money to actually do these kind of tests, build a pipeline, etc., etc. And this is just to tell you that here we are talking about treating sickle cell anemia patients. We're talking about tribals, the poorest people. How to take such kind of a therapy to such people? That's the challenge. And this means that this challenge has to be let's say taken through multiple rounds, and we began to explore the possibilities. We started all this work in 2017, okay, when I joined IGIB. And then along the way, we started negotiating with regulators, with agencies, with patients, with hematologists. Now, even at the level of the Minister of the Tribal Affairs, and try to see if we can push this all the way up till the point where now they are convinced enough, and we are happy to say that they have now actually funded this all for a clinical trial. This somebody sent me yesterday only. This was at the Union Budget yesterday, where the Finance Minister actually presented that they are trying to now build a national campaign for sickle cell anemia. Lot of people have come together to make sure that it reaches the right ears, let's say, for screening and evaluation. But most importantly, we have been funded for a 5-year kind of a project. It's a huge project by the Ministry of Tribal Affairs and the Department of Science and Technology, where this clinical trial for CRISPR will happen. And this will happen at All India Institute of Medical Sciences in New Delhi. We will use our technology to edit the cells. They would put it into the patients and see how this works. These are the type of editors that we are going to work with, which will be adenine base editors, which can simply sit on the DNA and correct the mutation. So I won't go into details so much. But it's a lot of people who are involved. So for example, here is a lab, Orko Shubhro Ghosh at Bangalore. He's making viral vectors where he can package these CRISPR components and put it inside. There are the NIH in Bombay who are studying what is the immune response to such vectors. And of course, IICT and University of Pennsylvania. You might not know this person, but you will know him in a year or two because he's one of the contenders for the Nobel Prize. His name is Drew Weissman. He is the father of the Pfizer Moderna vaccines, the mRNA technology, okay? And he was kind enough to come and make all these ENF and Cas9 RNA, using the same technology that goes into the vaccines. And we are currently testing this in preclinical mouse and so on. It's not just sickle cell anemia. We have also, seen that it has worked very well for other diseases. This is from LV Prasad Eye Institute in Hyderabad, where they use these kind of technologies to treat patients who have lost vision, for example. And this could be, they use the patient's own stem cells to correct it using CRISPR and then make it into a retinal pigment epithelium cell layer and then transplant it in the eye with the hope that some of the vision is restored. So this is also going into some kind of a clinical trial as well. But just to summarize, this just to tell you that it is not a very simple process to take healthcare to people, because we learned it the hard way. We learned when we did the Feluda, we realized that there are a lot of different things, pitfalls and lot of problems that happens because sometimes we do not have so much industry support for such kind of healthcare things in India. So if you had to do that from scratch, it would require coming together of so many different types of people who are regulators, who are politicians, who are scientists, who are clinicians and so on. And of course, be very transparent with resources and sharing. So, if you want to use any of these, tools, they are all on AdGene. You know about AdGene? This is a plasmid repository, but you do not have to go to AdGene. You just write a mail to me and we'll send it to you. So if you want to do your own genome editing experiments, for example, here in Ramakrishna Mission. So with that, I'd like to thank all of you. This is the most important slide because there's a lot of people to thank. This is not done by a single individual. These are done by very excellent students, all like you actually, and who are part of this team. A lot of labs around the world, clinicians, doctors, and different policymakers and so on, and different types of funding agencies who support this work. So with that, I'd like to thank all of you, and if you have any questions, please let me know. Do not be shy about asking questions, anything, okay? Yes, please start. Yeah.
Hello. Good morning, sir. Sorry, I think it's afternoon already. Good afternoon, sir. This is Debojyoti Banerjee from Department of Microbiology. I had a question that we are taking blood cells, we are editing it. We are editing it like for sickle cell anemia, as you have stated. We are changing it that GLU into valine, I guess, like in the beta globin chain. What we are doing with the Cas9. So after that, we convert it to stem cells. We make it into organoids, and then
No. So for sickle cell anemia correction, our blood has got its own stem cells. It's called hematopoietic stem cells.
Yes.
So we take out the hematopoietic stem cells from the blood, we correct it, and then we put it back into the patient. Once it goes into the patient, it goes into the bone marrow and it settles down over there and starts producing new cells. And these new cells are now corrected cells. So the patient receives only a single dose and is corrected forever.
But there are other hematopoietic cells as well in like different parts of like bones. We have in sternum, suppose, or for femur,
right.
we have different sets. Like so, if we correct maybe one of the stem cells, if we put in one of the edited stem cells in our femur,
right,
then what happens to the stem cells of
very good question. So what happens is that this difference comes because of the difference in the lifetime of a sickled RBC versus a normal RBC. So a normal RBC has a lifetime of, correct, 120 days. Does anybody know how long does a sickle RBC have? It has only 30 days. Okay, so this means that if you have a proportion of RBCs which are corrected and becoming normal, they will outlive the other RBCs, and over time the other RBCs, which are sickled RBCs, will get cleared from the body and you will only have the normal ones. So after 6 months to 1 year, the patient will be almost like he has been reborn. So there is no more sickled RBCs at all.
Thank you, sir.
So since these are corrected, therefore these are now corrected hematopoietic, and they will start dividing and making so a proportion around 50% will be corrected, and these 50% will give rise to more corrected cells. So they will divide as well, stem cells, right? So they will give rise to more corrected cells. Will get eliminated over time because you have a 120-day lifespan versus a whatever lifespan, and these stem cells are also dividing. So over time, this balance will ensure that only the corrected cells are remaining.
Sir, my question is, we have heard that RT-PCR test and rapid antigen test used in COVID detection. So how the CRISPR technology and Feluda helps for COVID? Is the same like RT-PCR test?
Yes, so it is different because your rapid antigen test, as the name is telling you, is an antigen test, which means that it looks for the COVID antigen in your blood, which is a protein. Feluda is similar to a RT-PCR test because it looks for the COVID RNA. Okay, so you can compare the Feluda with an RT-PCR, and you can the antigen or the antibody tests are essentially rapid tests. Also, you can do it at home. These are for your antigens that are present in blood. Okay, proteins.
Any other question?
Shout your question. Yeah.
Hello sir, my name is Shagnik Goswami. I am a student of Department of Botany.
Yes.
Sir, my question is the technology what you were talking about, you tested it with sickle cell anemia. What about cancer? Is any work done?
Yeah,
on
yeah,
cancer?
lot of work. So actually, cancer is a hot topic, na? So a lot of people are working on genetic engineering for cancer as well. And I don't know if you know about something called CAR-T therapy, which is one of those landmark therapies that are almost it's possible to cure cancer completely.
Is it linked with immunotherapy or?
Yes, it's a type of immunotherapy. So the way the CAR-T is made is done using CRISPR. So some of the companies are using CRISPR-based CAR-T therapies. So CAR-T therapy has come to India also. At this point, it is not affordable by everybody because it costs around maybe 40 lakhs or 50 lakhs. But it is a therapy which can get rid of cancer forever. But if the price comes down in the next few years, maybe then it will become much more affordable for the rest.
But is it possible to trace out the specific stem cell which is causing cancer, like you had done with sickle cell anemia?
You can do that. So you can cure the mutation which causes cancer. But I think what the CAR-T is promising is, because what it does, it takes from your blood your T cells out, which are T cells are the ones which are immune cells. And then it modifies them to tell them that, look, you're not going to be finding other cells, but you're going to search and find cancer cells in the body and then kill them. And then you take these T cells and put it back into the body. Okay, so then they go all over the body looking for cancer cells, killing them, looking for cancer cells. It's amazing technology. So that has been, let's say, very remarkable in the way it has progressed, and it is moving very fast. Yeah.
Okay, sir. Thank you.
Hello sir, I am Riturik Bhattacharya from Department of Microbiology. So I have a question that during the embryonic development, there is a phenomenon of random monoallelic expression. So that helps in the fate of the embryo in a matter of about 14 days. So how can CRISPR systems help in the eradication of those embryonic diseases or fetal disorders?
They can, but at this point of time, there is an international moratorium on not using CRISPR in embryos. You know about a Chinese scientist, He Jiankui? Yeah. So he tried that, of course, and they went to jail.
Yeah.
So it is more or less, let's say, understood by the scientific community all across the world, that till this protein becomes so foolproof that it will not cause off-targeting, embryonic editing will not happen. But adult editing, like in diseases, can be allowed.
But if it's a congenital disease, is it very, feasible to edit in a...
It is feasible. Definitely it is feasible. But it is more like, if you try to do that, then maybe somebody wants to do it for a congenital disease. But then somebody might say that, well, I know that there are some genes which causes higher IQ. I want my baby to have a higher IQ. How will you control it? So you will have people who can pay more will get brighter kids.
Okay.
And that is not good for the society, because the society thrives on admixtures, on diversities. Okay, if this diversity is lost and there is a selection pressure due to artificial genetic engineering, then the society as a whole will be prone to diseases which are unthinkable of. So that is why embryonic genome editing at this point is not done.
Thank you, sir.
Any other question? So if
Good afternoon, sir. I am Atmadeep Ganguly from Department of Microbiology. In regard to the previous question, like he asked for cancer, my question is about Mycobacterium tuberculosis. Like we know it can also survive in hematopoietic stem cells. So can CRISPR, this technology used to detect those particular stem cells and like it can help in the eradication?
Yeah, of course. So Mycobacterium TB is, of course, it's a DNA bacteria. So you can use the CRISPR to eliminate TB. So you could chop off the TB genome and essentially stop it from replication. The question is, how do you deliver it to the right tissue? Because it's in the lungs mostly. So you need to find a delivery route for CRISPR to go safely into the lungs, and then you move to the cells where the TB is, and then in those TB cells, you have to go and chop the DNA. So there are people who work on that, but it's not a very straightforward thing, because the delivery route is the most complicated thing. Can you take maybe a nasal spray or something like that? Yeah.
Thank you.
So if there is no any question, so I have one question.
Sure, sir.
So, actually, in plant science, in GMO, there are lots of restrictions in releasing the GMO crops. And particularly for the genome-edited crops, this GMO legislation is not considered because it's not typically the GMO. So it's like mimicking the natural mutations. So what about the application of the CRISPR in the human therapy or therapeutics like that? How is the legislation?
So for human therapy, you mean?
Yes, yes.
So human therapy, the legislations are evolving. Like, it is, India has not had a legislation because a product was not there. Now that we are moving towards the first trials, so that is why some of us are actually in such committees which are making these kind of legislation so that, it moves along with the scientific field, that they are not going to stop it. Of course, they are not stopping it because there is a lot of encouragement to go in that direction, knowing the power of the technology. Plants, of course, this was now categorized as SDN1, SDN2, and then it has now gotten approval. So our CRISPR, the ones that we developed, the ENFN Cas9 have now been tested in brassica, in wheat, I think, and also in rice and some other plants. And they are planning to also see how to release this officially for certain types of traits and so on. But for human diseases, it is on a case-to-case basis. Yeah.
We'll take only one last question.
Morning, sir. I'm Ruktim from Department of Microbiology. Just like you said about cancer and everything, can you highlight how it can help in the treatment of HIV, or even if it can?
Yeah, HIV is one of the top targets for CRISPR because HIV works because the viral RNA has incorporated into the human genome in the form of DNA after reverse transcription.
Yeah.
So there are people who are trying to use the CRISPR to cut this chunk off from the DNA. So that has gone into, I think they have finished the preclinical studies. Now they are going into clinical trials also. But it's a very hot topic. So many people are trying to do that. It's very powerful technology because if you can play with the genome, you can essentially do a lot of things which was unthinkable of. So HIV is definitely one of those targets.
Thank you, sir.
Thank you. We'll not take any more questions. If you have any question, you can freely interact with the speaker later on.
Thank you very much. Very nice questions. I really enjoyed a lot. Yeah?
So now I request our controller, sir, Swami Vedanuraganandaji Maharaj, to give the vote of thanks and end the session.
So it was really a very enriching session from Dr. Debojyoti Chakraborty of IGIB, Delhi. So we have seen how varied his career is, starting from genomics to music. So while I was hearing his talk, it was as if that as in music you have various notes, and with various combinations you have various ragas. If you just change one swara, like a Shuddha swara to a Komal swara, then the raga changes. Then again if you change it, it will again revert back to a different raga. So maybe he is so much musical in his approach that genomics and genome editing is also something like changing the raga and again playing it back to the same raga. Maybe that type of an editing process is taking place even within the gene. So we are really thankful to Dr. Chakraborty for his wonderful talk, for his enriching talk, and we are also looking forward for his another wonderful session in the afternoon with Sitar recital by Dr. Chakraborty. So on behalf of Ramakrishna Mission Vivekananda Centenary College, Rahara, I extend an heartfelt gratitude to Dr. Debojyoti Chakraborty on behalf of the Department of Botany and DBT STAR College team, which has organized this seminar. We are really thankful to sir. We are really thankful to Dr. Chakraborty's parents as well for coming over here and by their gracious presence and blessings to all of our students. And we are thankful to all the teachers and students for their participation and enthusiasm. We look forward for such lively academic sessions in future as well. And finally, at the end of this session, we will again meet around 2 o'clock for Dr. Chakraborty's Sita recital session. So all the BSc first semester students of all the departments